Cattle are very diverse. Cattle are raised all over the world in many different climates. Typically, we have a limited number of cattle breeds that are used for production in the US. So as climates change, there might be specific traits from other breeds around the world that farmers would be interested in utilizing. One thing that improved genomes allows us to do is to more accurately bring traits in from other breeds that we might be interested in.
Genome assembly technologies come a long ways. The process used to take millions of dollars utilizing large international consortiums. Dr. Rosen now assembles genomes much faster for a fraction of the cost, thanks to technological advances.
If you're trying to assemble a genome, you can think of it as a very complex jigsaw puzzle. And when those pieces are really small, it can make for a very hard puzzle to put together. And now we're able to do multiple genome assemblies within a single lab that are orders of magnitude more accurate, better in every way than what was typically done by these very large consortiums.
Advances in technology have yielded many research benefits, most notably T-to-T or telomere-to-telomere assemblies.
The first cattle genome had, I believe, hundreds of thousands of regions where information was just missing, known as gaps in the genome assembly. But now, with the combination of highly accurate and long sequence technology, we're able to assemble genomes without any gaps. The ability to go from the start of a chromosome, which our chromosomes are capped with telomeres, you go from one telomere all the way through that chromosome without any gaps to the other telomere, and that's what we call T-to-T, telomere-to-telomere genome assemblies. Instead of 100 base pairs, you can sequence and look at 10,000, 100,000 base pairs at a single time, and now you have the ability to span some of these more repetitive regions and to place those sequencing reads to put that puzzle together. So that one turned out great. I think we probably need one more flow cell data for this guy, but then I think we'll be all set. Once we have the genome of an organism fully sequenced, fully assembled, and as correct as possible, now what researchers are able to do is to understand at a very fine scale what these genes, what these instructions are doing in the genome. The better our genomes get, the more traits we're able to understand. Now with our improved genome assemblies, we're going to be able to expand that to more regions of the genome, expand that to more traits that people might be interested in, which would enable us to improve production in ways in which we previously weren't able to. It's going to open up new avenues for improving animal health and animal production.